So today's speaker is very special in terms of he has a unique achievement for studying pain biomarker. So Professor Wu Chung-wan major research is integrated brain model of human affective experience and clinical outcomes, especially in neural mechanisms of pain and emotional dynamics. And he has been assistant professor from Sungkyunkwan University and also Institute for Basic Science since 2017. And he got a PhD from Psychology and Neuroscience of University of Colorado at Boulder. And at the same time, he's done postdoctoral fellowship from there. After he's got bachelor's degree from Biological Sciences of Seoul National University. So let's welcome Professor Wu.
Thank you so much. Can you hear me?
Yes, you can start.
Yeah, okay. So thanks a lot for having me here, and it's a great. I think I saw the previous speakers, and I'm really feel honored to be on the website on the same colloquium with them. So today I'm going to talk about a little big picture of how to use this fMRI, functional neuroimaging, especially fMRI, for decoding pain and emotion. So can you see my screen? You can see, right? Okay. So it's not an exaggeration to say that it's an era of brain science from United States, Europe, and China, Japan, and even in Korea. There has been a lot of big kind of brain initiatives to support the brain research. And from the small brain to large brain, there have been a lot of advancement in this in vivo neuroimaging methods. Among them, I think MRI really made a really great breakthrough in studying human brain. Especially, it provides a really quick snapshot of a functional kind of activity, and also the structure. We can relate them with structural changes, and so that we can that opens connect the brain with mind and the body. And also, thanks to the MRI, that really combines all these different disciplines that existed separately previously, and kind of created a new field of area studies. And actually, I'm a part of these new areas that has a really integrative and interdisciplinary approaches. And as you can see, the fMRI, especially combined with machine learning, made a really great progress in decoding things, our thoughts and emotions. And here you can see the left side is decoding what we see. That is already done 10 years ago. And the right side, it shows the decoding what we dream. It's also like around 8 years ago. All these thought decoding approaches are really promising, and this is a more recent one that from the Kamitani group that I really liked, Yuki Kamitani from Kyoto University. He visited us around that time, the 2017. And anyway, so we really like each other's study. But anyway, they are doing really well, and then they use the deep learning and combined with fMRI data to reconstruct what we see. But at the same time, there has been a lot of kind of warnings that the brain science is, for example, brain science is drowning in uncertainty. And also there has been a paper that shows many of the fMRI findings has been false positive, and that was introduced in Korea as well. And also in the clinical area, there has been a large amount of complaints. It's ongoing, I think, because there is no one application for clinical kind of use. especially using fMRI. I mean, there are tons of applications using structural MRI, but not fMRI. So, and also from the cognitive scientists, they are asking this question: how can functional neuroimaging really inform cognitive theories? And many of the cognitive science researchers really think that maybe just behavioral level analysis and data are going to be enough. So kind of we are seeing the troubles in paradise. I can see the paradise in the future of fMRI for example, decoding of accuracies, but also there are many troubles in this paradise. So I will talk about these three things today. The first one is interpretation, and second one is replication and translation. Let's start with the interpretation. So let's say you got this brain map after the statistical analysis, and how can we interpret this activation map? So usually many of the fMRI studies papers really report many brain maps like this, and then they interpret these activation significantly activated regions and interpret this in a way. And I can explain the common way, conventional way of interpretation of this activation to you. So for example, they start with this local mapping of brain function relationship. So for example, they have participants do some sort of some specific tasks they want to know. That's a well-known task with a well-known function. So for example, pain or pleasure, they have participants do these type of processes, involve these processes, and then they observe this local brain activation, and they do just local mapping of this relationship, and then they do another task that is not well known, so kind of task of interest. So they wanted to ask some new function, and they want to connect this A and B. So I will, maybe it's unclear. I will give you some examples. For example, as I said, the pleasure is kind of really a basic ingredient of our human experience, right? And then they do some task in the scanner, and they observe this, for example, nucleus accumbens activation. And it's well-known, kind of well-known connection between this pleasure and nucleus accumbens activation, actually. So they have this connection first, that is a local mapping. And then what they did in this study, the Tamir et al. 2012, is they asked participants in the scanner to talk about themselves. So talk about, kind of disclose themselves, right? And they found this self-disclosure is also activate the nucleus accumbens, and they are connecting these two. And as you can see in this title, disclosing information about the self, that is a self-disclosure, is actually intrinsically rewarding. So you can see this logic, right? You have this local mapping of the basic function, mental function, and then you do the task that you don't know well, and then they kind of establish the mapping between these two mental functions. And you will be able to find many of the examples of like this. So for example, in this paper actually got a lot of criticism, but at the time people kind of interpreted the dorsal anterior cingulate cortex activity as an emotional conflicts. Some complex processing is kind of important role of DACC. People have been saying that, and then in this specific study, they showed the pictures of presidential candidates at the time, the United States presidential candidates, and then they saw the Hillary when they showed this Hillary Clinton's picture to participants. This DACC region showed activation, and they interpret that as emotions about Hillary Clinton are mixed. So this was actually introduced in New York Times. So this type of logical kind of reasoning is everywhere. And this is another paper from kind of, it's old now, but 2003. And that was same kind of really sensational at the time. So people also knew that the pain really activates dorsal anterior cingulate cortex. And in this paper, what they did is that they excluded someone from the game. So they called it social rejection. And then they also observed the DACC activation, and then they interpret that as a rejection hurts. So this is a really famous paper. And then actually based on this paper, they kind of build a whole field of social neuroscience. And this book is also translated in Korea. So this is Matthew Lieberman's book. And then actually Naomi Eisenberger and Matthew Lieberman, they are big names in social neuroscience, and they are actually got married anyway. They are still professors at UCLA. Anyway, so these type of analysis and reasoning interpretation is everywhere. However, how tight is this relationship? Because as you can see in this reasoning, is this logical reasoning, the relationship between this pain and DACC, that what I mentioned about, like the local mapping between brain function relationship, this is the foundation of this reasoning. But how tight is this association? And to answer that question, we need to consider two directions. So one is pain to DACC activation direction, and the other one is DACC to pain direction. But I will talk about this direction first. So when people experience pain, what is the probability of seeing this DACC region's activation? And actually from our lab's data, we kind of collected a lot of data from people while they are experiencing pain. After we kind of applied a really stringent threshold, we can see the dorsal anterior cingulate cortex activation. That means this direction is correct. So when people are feeling pain, you are going to see this DACC activation. But we have to think about how specific is this going to be, because it could reflect non-specific mental functions like aversiveness or arousal, attention, salience, sensory process, or action plan. Because if you experience pain, you want to do something about that. So all these different aspects, different components that is not actually specific to pain can also influence this activation. So the more important thing is actually the probability of being in pain when you are observing the DACC activation. And to say this, the high probability of observing pain given DACC activation, we have to rule out this activation is about action or salience or these other non-specific things. But when we are actually analyze the data from previous studies, so this is an automated meta-analysis tool called neuroscience.org. That is actually from the lab that I got my PhD. The Tor Wager is my was my PhD advisor, and Tal Yarkoni was a postdoc when I started my PhD. And the Tal Yarkoni is I think he, in addition to, I mean, as well as my advisor, they are, I think, geniuses. And what they did is that they actually build a text mining kind of model program that kind of automatically analyze the previous studies, and they kind of and also read out the activation table. So because it takes so long time to do these meta-analysis manually, what they did is using text mining technology, they make a big table that activation coordinates and also the words, the word frequency. So based on this, actually what this can provide us the cool thing is we can actually calculate the base rate of activation. So probability of seeing activation of each region, regardless of any studies. So this is the map. This map is from this paper, and at the time they analyzed more than 3,000 papers, and this is the probability of seeing the DACC activation, no matter what the paper is about. So that means this, as you can see, the DACC region has more than 20% probability. That means if you have 100 papers, neuroimaging papers, and if you randomly pick a paper among them, and then 20% of, I mean, as a with a 20% probability, you are gonna see the DACC activation, no matter what the topic is. That means this DACC area has really low specificity about the mental function. And so that is the actually there was a really interesting event or occasion in this field. Some people, this Matthew Lieberman and Naomi Eisenberger that I talked about, and they actually use this neuroscience meta-analysis database to argue that this DACC area is really selective for pain, specific for pain. And then there was an answer, like the really pain in the ACC, like is ACC really specific for pain? And then so many people, including Tor, my advisor, and Tel Yarkoni, they wrote replies to say that no, pain is, the ACC is not specific for pain. So this figure actually I also kind of contributed to making this figure, because what we did is we collected the paper that reported this area, the activation of this area, the DACC area, and then we actually manually go over the papers and then write down the topics. And as you can see, there among all these papers, 12% of papers was about actually pain, but more than that, like 20% was about attention and cognitive control, and also many of, I mean, these many other terms has been related to this area, the activation of this area. So after that, 2016, there was a news reviews in Nature Neuroscience about DACC area. Actually, the title was really funny, and that was a Rorschach test for cognitive neuroscience. So why that's a Rorschach test? You might know about Rorschach test or not, but Rorschach test is a projective test, and actually it's been used in psychiatry in Korea as well. Still, it's been using, and if you are going to psychiatric kind of hospital, I kind of got trained there. I has been clinical psychologist trained in hospital for 3 years, and then this Rorschach test was one test that I administer directly, and then as a examiner, we ask patients about what do you see in this picture, and then they see each person see different things. So some people see some bats, or other people see faces. Anyway, so the whole point of this test is a projective test. So by showing this really ambiguous figure, we are getting information about the person. So I wanted to share this quote. So it sometimes seems like a dorsal anterior cingulate cortex is a Rorschach test for cognitive neuroscience. We all see something different in it, and what we see may tell us more about ourselves than this DACC. Then about the function of the region. So what this means? This means that attention researcher will see attention in the ACC, and pain researcher will see pain in the ACC, or cognitive control researcher will see cognitive control in there, and language researcher will see language in there. So the interpreting this DACC activation based on their, we like interpreting the results based on this DACC activation has a problem. That's a crisis. That's the issue, the specificity issue. So how it can go wrong? Actually, it's another funny story that after the Matthew Lieberman and Naomi Eisenberger published a paper on Science about social rejection really hurts based on this DACC activation, psychologists really got really excited, especially I love these psychologists. I love these people's work, but they were really excited about this really the physical kind of root of this social rejected feeling. So there has been some papers about review papers about social pain theory, but that didn't stop there. Actually, love hurts and also math hurts. All these papers are based on this DACC activation. So the interpretation based on this DACC activation is kind of intrinsically flawed. And if we really analyze all the words that activated the DACC, everything hurts. So seeing Hillary Clinton also hurts because, as I said, the previous that study interpreted this DACC activation as an emotional conflict, but it was actually we can also interpret that as a pain, right? So seeing Hillary Clinton may hurt. So what I want to say, the point that I wanted to make is establishing tighter associations between the mind and the brain is really, really important. And throughout my PhD and also in the 2017 paper, I made this really clear that we have to apply some criteria to say that we established a tight association between this mind and the brain. What I mean by brain is a brain measure, some activation of regions or patterns, whatever the measure that we have. So the first one is the first direction I explained, pain to brain activation. For example, I'm using pain as an example. So there is in other words, it's a probability of observing that brain measure given the pain experience, and that we call that. That has other names like sensitivity or forward inference. But many studies just stop there and interpret their results. But we have to go further. The second one is large effect size between pain and brain measure that we can measure with Cohen's d or explained variance. And also we have to test the specificity. So there is the other direction. There is when there is no pain, what is the probability of no brain activation? As I said, if there is a high base rate of the brain observing that brain measure, this specificity should be really low. And combining all these measures, we can say the probability of pain, the person is being in pain given the brain measure observation. So there is a basically positive predictive value or brain decoding. We call it also as a reverse inference. So to meet this all criteria is not easy one, but we have to establish this first and then interpret the results. But people are not doing that a lot. So that was like my message, strong message that I wanted to make to the field at the time, like a few years ago. But with the traditional mapping approach, so-called like statistical parametric mapping, it's not enough for that. Because in traditional mapping approach, we usually what we do is we have participants to do some task, and then we test whether this effects of task or function is observed in each region separately. So the question is there an effect at each voxel or each region? So that is actually aimed for local mapping. So there is no model for distributed representations, but we know that the brain really has the distributed representations for something. So many-to-many mapping is actually what we have, but there is nothing for that in this approach. Therefore, we can actually we cannot really achieve a large effect size. So we ended up having really small effect sizes, and also given that we have to test we have to conduct a lot of tests, we are ended up doing these massive multiple comparisons. So we have to correct for the multiple comparison, and then the power getting really low because the threshold is getting high. I will explain that again in later slides. So our idea at that time was the reversing the equation, and I think this is becoming more and more common practice in the field. So we call that as a predictive modeling approach. So what that is doing is the question is different. Can we model joint contributions of multiple brain regions for the outcome? So we need to combine all the information distributed across the whole brain and then model one thing. So that is basically the brain-wide pattern-based mapping and modeling, kind of actively modeling the distributed representations. And therefore, given that we are incorporating, utilizing all these information across the brain, actually we can achieve large effect sizes. And there is no need for multiple comparisons because we are building one model for one outcome. So just one test. And therefore, we can have really high power. And also, given that this is like basically the pattern recognition task, and we are incorporating all these information from multiple voxels, so multi-voxel analysis, basically, we can filter some indirect connections, and also we can identify masked effects. So basically enhancing specificity and sensitivity of detecting signal in our region. Also, the procedure, usual common procedure of doing this is training a model, kind of pattern finding procedure across whole brain, whole sample, or we can do this each sample if we have enough data for one subject. And to enhance the neuroscientific kind of interpretability, we do some bootstrap tests or some occlusion tests, whatever tests we are doing many kinds of tests to interpret this map. And then after we get new data, we just do the pattern, calculate the pattern expression value using this predefined map. So these are all new data, and this is the model we have. And then by doing the dot product, we can actually calculate the pattern expression value and then predict or classify the outcomes. So this is basic procedure, and I'm going to explain you, give you some examples, the example studies that we did with this approach. So this is our, the first paper we used this approach, and that was used to decode the pain. So basically what we did is we modeled this whole brain to predict this pain rating, and this is the map that we got. And this is the thresholded map after bootstrap test, but actually there was pattern. So each voxel has the predictive weights. So each voxel has a value, and all these voxels jointly predict the pain rating. So all this information is being used. And we named it as a neurologic pain signature, and we call it NPS. So this NPS model was able to discriminate physical pain from non-painful warmth and pain anticipation and emotional pain and pain recall with a high sensitivity and specificity across 4 studies. So people really like that. And then also this red line is the NPS response. So basically we calculated one score from one map, and then this shows the decrease of MPS response to the remifentanil analgesics. So this is the drug concentration, and you can see really nice curve decrease that is really looks similar to the drug concentration. So we can see the brain response actually decreased response to the analgesics. And after that, after we developed that model, we actually use that again and again in a new data set to test, to establish this these things, like what really activate these MPS. So what how can I say, the MPS is activated by what, which condition. So actually the MPS is developed using this noxious heat, but actually it was activated by noxious pressure or electric shock, and also other types of visceral and other types of somatic pain. So we now know that MPS is actually respond to many different types of somatic pain types. And also we have shown that the MPS is not activated by aversive images or social rejection. This is a like direct kind of rebuttal of like the dispute of the for the Naomi Eisenberger's paper, that okay, our MPS is not activated by social rejection. So maybe social rejection is not gonna hurt. I mean, it's not, I mean, it hurts, but different way. I will explain that in a minute. Anyway, and it's not activated by observed pain. This is another story, like whole story, that people have shown the pain empathy activated this DACC, and they interpret that as a, okay, seeing others' pain actually really literally hurts, and then maybe it's not the case. And also like placebo effects, it's not activate the MPS. So all these things really show that the MPS has some specificity about pain. And so the next study that I did is comparing this physical pain and social pain, actually focusing on social rejection. And what we did in this study was we showed, we give the heat. So this is heat pain apparatus, so equipment. So give the hot or warm heat stimulation to people, or we show that friend's photo or ex-partner's photo, because we recruited participants who recently experienced unwanted breakup. That is a like 차인 사람들을 부른 거죠. So they experienced unwanted breakup, and then they had to see the ex-partner's photo in the scanner, so they feel really hurt. So when we analyze the post-scan description of the stream of thought during the ex-partner trial, they actually felt sadness or anger, and also they used pain-related words in 10% of people. I mean, 10% of reports. So like painful, got hurt, and feeling painful, like that. And with the traditional mapping approach, that is actually the same approach they used by Naomi Eisenberger group, we were able to see the DACC activation and also anterior insula activation. And interestingly, we can observe the secondary somatosensory area and also dorsal posterior insula. We know that this S2 and DP insula is actually the regions that are kind of really specific for pain, respond to pain really specifically. So okay, only with the activation level, we can see that there is a really overlap between this somatic pain condition versus the social rejection condition. So this is actually this finding can be used as a foundation of the social pain theory. But we didn't stop there. We used the predictive mapping approach. Basically, we used a linear support vector machine classifier to obtain some specific multivariate fMRI pattern for pain and rejection on the same data set. And we were able to develop a marker classifier for somatic pain, and also we were able to develop classifier for social pain, social rejection, and they were not that correlated. and each marker, each classifier actually was specific to classify each condition. Like the somatic pain marker classifier was specific for pain, and also the social rejection classifier was specific for ex-partner condition, the social rejection condition. And also, when we see the patterns within each region actually had the similar level of activation, their patterns were quite different. So no one region showed significant correlation. So they were all different. So the message, the point of this paper was, even though the pain and social rejection activated the same region, like the ACC, they are represented in a really distinct pattern. So patterns were different. So maybe the pain and social rejection could be different. So if you ask me about, does rejection hurt? Maybe it hurts, but differently. They are different. But this has a really important implication, because if you can see this title, feeling the pain of rejection, try taking a Tylenol. So people started to actually consider prescribe the anti-pain drug for the social rejection. So maybe not, but the problem is we don't know well about the mechanisms of Tylenol, and also maybe anti-inflammation drug like Advil, maybe that works for both, maybe pain and social rejection. Anyway, the people started to think about this option based on that paper. So that science paper, 2003 paper. So we need to be really careful about the interpretation of brain activation maps. And the same conclusion for social empathy. So we actually found the social and vicarious pain, that is pain empathy, the seeing others in pain, they were represented by dissociable pain, multivariate brain patterns. So this is first trouble. So that as I showed you, the local mapping, so the mapping between the brain measure and the function is actually very weak than you thought. So it's imperative to establish tight associations between brain measures and outcomes first, before before doing further interpretation. And therefore, pattern-based predictive mapping approach provides a promising direction to develop this kind of some specific and sensitive markers. I mean, this is the longest one, and the second and third will be short. The second trouble is replication. As you may know, there was a really there were many papers that saying many of the published findings are false, and like there is a reproducibility crisis. So like this reproducibility crisis, and then more than 90% of scientists thought there is a reproducibility crisis like 5 years ago. I think the issue is still there. And there are so many things that contribute to this replication crisis, but I will talk about a few things. And the first one is this one, the winner's curse. This plot is from this 2013 Nature Review Neuroscience paper, and I will explain one by one in this plot. So here the x-axis is statistical power. As you might know, statistical power is about the probability that it will correctly, my test will correctly reject the null hypothesis when the null hypothesis is false. And with a large number, I mean the high sample size, large sample size, and if there is a high signal to noise ratio, that is basically the effect size, or if we apply the low threshold, we can get high power. Why the low threshold? Because this statistical power is the false kind of what is that? False negative, basically. So so when you are applying the low threshold, you can say the yes to many things so that you can actually correctly, there will be, you can correctly reject the when the null hypothesis is false, but also when you are applying the low threshold, false positive also got increased. So there is a trade-off between this statistical power and false positive rate. But anyway, the statistical power is the focus of this discussion. So as I said, the power is really low in psychology studies, generally low, because the people's behavior are really noisy. So basically the fMRI study, really, we really want to study the human behavior in the scanner. And so the effect size is intrinsically really low. And in addition to that, we have the fMRI noise, measurement noise, right? So the behavioral noise plus fMRI noise may give us the terrible power, terrible effect size for that. The reason is sample size, for fMRI studies, usually we have really low small sample size, a small number of subjects because it's expensive. And as I said, low effect size. And if you use the conventional, traditional approach, you are gonna have to need to do the multiple comparison correction. So therefore, the threshold got increased. So because of these three factors, you will get the really low power. So remember that fMRI studies have really low power, and the here y-axis is a stronger results than real. That is bias. Actually, there was a voodoo correlation discussion back in around 2008. But anyway, there is a stronger results than real, and the relationship between these two factors is negative. So basically, if you have really low power, it is likely that the results are biased. Biased findings, because given that all these published results are positive, kind of biased results, because you only report the significant results, right? So you just throw away the non-significant results and then report the biased results, and therefore the most of the findings are false positive, basically. Then you are going to get replication failure. So that is the first factor, important component, important contributor to replication failure in the fMRI studies. And the second factor is flexibility in analysis. So people usually say that their hypothesis is amygdala activation. So they formulate their hypothesis as a region name, but amygdala has more than 1,000 voxels. And therefore, there are tens of thousands ways of a combination of these voxels, so that the amygdala activation as a hypothesis introduced some wiggle room in kind of interpreting this hypothesis. So it's really similar to this paper. So false positive psychology that was published in 2011, but we kind of got the idea, then we wanted to say false positive neuroscience because of this flexibility, and we actually published a paper with kind of borrowing the title. So false positive neuroimaging, undisclosed flexibility in testing spatial hypotheses allows presenting anything as a replicative finding. Anyway, so these two factors, and I want to argue that the pattern-based mapping approach, predictive mapping approach, is really good solution for that, because pattern-based marker development has a better effect size, as I said, because modeling all these information distributed across whole brain, it has the high effect size, and also it doesn't need to be corrected for multiple comparison because it is one model, so one test and done. And we started to treat these pattern maps as a research product. So that as a one assay, one model. So therefore, there is no flexibility in defining testing model. So if you want to, for example, if you want to test the MPS model that I introduced, in another data set, you cannot really change anything. So there is no flexibility in defining this spatial model. And also easy to test on new individuals and data sets. Therefore, we can actually test across studies and sites. So basically, this was the map that we made a while ago, the MPS test map. So after we developed the MPS map, the pain marker, that has been tested on many data sets across the whole world, and used for establishing this MPS's receptive field, and the MPS has been patented. So it's a new way of thinking. Brain map, brain model, just the weights that can be patented. Okay, and so we are doing actually this practice a lot now. And there has been other pattern signatures. We recently developed a connectivity-based marker for tonic pain. Early this year, we published this paper on Nature Medicine, and then we actually applied the patent in Korea and also United States. So you can actually treat these maps as the product. Okay, that's the idea. And so we really had a vision that there should be multiple candidate models, and and through the competition, we have to get the promising models that works across the whole the population level. So only with this way, we thought that we can actually get a really good biomarker for using fMRI. And we have to really share the models. So there has been given that based on the consensus that we have to share data, there has been many data sharing platforms, but not many model sharing platform. So I think that's a kind of intrinsic challenge, because sharing model, people don't like it. So there is this toothbrush problem, the kind of really interesting name. No self-respecting person wants to use anyone else's. This is about models. So they only test their own models again and again, and not interested in others' model. So we have to overcome this fear or this kind of aversion. So that we need to share the model through this GitHub or Figshare. Okay. And actually, after we developed this tonic pain marker, we shared that model through the website. There is a Google form. If you want to test our model, you can fill out the Google form, and then that goes to send the mail to our school about this mutual confidentiality agreement. And then if you sign it, and you can get the model, basically. Anyway, so for replication, multi-study, multi-site effort to share and test pattern-based neuroimaging markers will help replicability in neuroimaging research. And the last part is the translation. As I said, there has been a lot of complaints from the clinical field because there has been no kind of really real clinical use of these fMRI models. And as you might know, Tom Insel, who was the previous NIMH director, who he left for Google, and he's in the startup company called Mindstrong. And this is from interview. I spent 13 years at NIMH. I think I succeeded at getting lots of really cool papers. I don't think we moved the needle in reducing suicide, reducing hospitalization, improving recovery, and so on and so on. And anyway, he felt responsible for that, and then he left for a startup company. Hopefully, he can develop new things, but still, based on fMRI, I guess many of these large costs spend on fMRI studies because it's expensive, but we haven't been able to get any one application. So actually, I did some kind of survey. I did a survey like back in 2017, whether about the studies that use the pattern recognition methods for translational purpose. And then it got really increased, and it's getting more and more popular now. And I kind of looked at the use of these markers, and this shows the proportion of pattern recognition studies for each diagnostic categories, and this is the use. And I want you to focus on this part. People have been used their model, so they developed their model only for diagnosis, not only, but majority of them. And I discussed with clinicians with these results actually at the time, and I kind of had the question whether these diagnostic models based on neuroimaging will be really clinically informative. And I kind of saw, my answer is maybe not, because there is already existing diagnostic test, and what will be really useful then? So I met this article that is from Harvard Business Review about the machine automation, and they said that the automation map has two dimensions. One is predictability, and y-axis is risk. And if I take some example, like for example, the driverless car, actually it really has a high accuracy. So predictability is really high, but hasn't been used as a full kind of autonomous drive now, until now, because the risk is also high. But the credit card fraud detection is the predictability, the accuracy is really low, but it's been used in the maybe you got a lot of messages. I mean, when I was in the United States, I got many messages from the cards company that, oh, okay, your card has been used in South Korea. Maybe the fraud is, so the fraud detection is happening now because the risk is really low. And only this part, they say the automation will be happen. And I think when I saw this, okay, for the neuroimaging marker, we might need one more dimension, that is clinical utility. So I kind of placed each dot myself, but maybe the diagnostic test, as I said, people have been used the machine learning for developing diagnostic tests based on the functional neuroimaging a lot, and then accuracy has been really high actually, like 90 for the some diseases that's around 90% accuracy. But you know, the clinical utility is really low. The clinicians really think that, I mean, we diagnose. I mean, they already have the diagnostic test and they diagnose, but what is the added value of this marker? So there's nothing. So because of that has really low clinical utility, maybe that is not going to be used in the clinical clinic. But for these, like early detection or predicting treatment responses or risk assessment, all these are really hard decisions, even for clinicians. So maybe focusing on these areas, even though the accuracy is low, may help us to really make a really useful biomarker, neuroimaging biomarker, and clinically useful tool using neuroimaging, functional neuroimaging. So in this 2017 paper, I made a clear point that we need to focus on this area rather than just diagnostic model. And one more point is we really need to use the independent test data for the model testing. So this is a classification performance for model development sample. That is, most of them are cross-validated results. And as you can see, the accuracy is kind of okay. And if you compare with this prospective testing, this is the testing the model on the whole new data set, like unseen data. and then the accuracy got really low, decreased. This means there is a bias, basically. And the large data set, I mean, maybe people say, okay, I didn't use this prospective testing approach because I have not enough data. But it was not the case, because many of the studies that have no prospective testing used the consortium-based multi-site data, like EDNI type of data. So I found that the people just didn't do the prospective testing because they thought this is not important, or maybe that didn't work. So I made a clear point that we should have the independent test data set always. And if you see, if we locate the models in this framework, we can find the most of the models is actually stuck in development stage and not many models there. But there were some promising models that can maybe translate it into the clinic. And we made a table using those promising candidate markers. And I found one thing that, okay, they have names. And I was like pondered about that, and then realized that the naming markers has a philosophy that they are gonna use. That has an intention that includes the intention, researchers' intention, that they are gonna use this marker again later. So after I realized this, I started to name all these markers. And as you might remember, we name all these things. And then the tonic pain marker is, we named it TOPS. So we are going to keep using that again and again and test on independent data set. So for the translation, we need to develop clinically more informative models, and also translational neuroimaging models should be easily applicable to new individuals and new data sets, accelerating rigorous prospective testing. So to enter the promised land, we need to be ready to develop specific and sensitive brain markers and conduct multi-site studies, and build up data and model sharing platforms, and establish a brain-based biomarker development and validation process. That is not one shot, one study game. It's a really long process of developing markers. Okay, so that's all for that. With that, I want to thank my lab members and thank the funders. Thank you for listening.
Thank you. Thank you so much. I think this is a fascinating talk. Yeah, it was very interesting to start with the background. I think Dr. Wu did a great job for explaining and about the controversy and issues in the field. So I think this is a good time for asking question and discussion. So those who have a specific question, I think you can write down through the channel, which would be easier for the speaker to see. At the same time, I think it's a good idea to just ask, also discuss question with Dr. Wu. So anyone has any question from, let's say, yeah, maybe from Eunice?
Well,
yeah.
Let me start, and let me then start. I thank you, Professor Wu, for your very stimulating and elaborative talk regarding the subject. And I have just one quick question to ask is, well, what's the difference between paradise and promised land? In the beginning of your slide, you start to talk about a paradise, and the conclusion slide, you talk about a promised land. Is there any difference between those two?
What I meant by paradise was like the dream. So we are envisioning the future of the paradise, but we haven't been there, haven't got there. So I kind of rephrasing that as a like promised land, like we have to enter that, like just I'm using the analogy of like Bible kind of thing.
I see. All right, good. So my one technical question is that your MPS, like basically pattern, right? Pattern maps of fMRI voxels.
Right.
And but I know that we are, I mean, it's becoming the high resolution FMR and the high field are getting, you know, the popular, and basically a lot of people are toward that direction. And I was wondering if this pattern map, that model you generate, will, I mean, how significantly will it change, or even, I mean, increase like this sensitivity, or even decrease, right? Because like the point of going higher resolution fMRI is basically getting better resolution. But if you go push it to better resolution, then now you are troubled that your like a draining vessel bulb and this micro vessel bulb may be separated, and this pattern map could be changing. I don't know, but so is this pattern map will be universal, or do you think it's just acquisition dependent? That's one thing that, because, you know, I know you are doing like a multi-center study, but I mean, we gotta really be careful about what the resolution of acquisition, field strength. There could, I mean, both contrast can have many contributions for many things, right? So I was wondering, do you think in this promised land, this pattern map will be universal, or need to be optimized? That's my just very, I mean, very broad question, but
no, that's a really great question. And actually there was a part of my my Shinjin grant actually. So there is a really important kind of consideration that I think if I have to choose these two, like the universal versus optimizing, I would choose the optimizing one. I think to be more accurate, I guess it should be optimized in some way. But before that, before we get the better kind of figure out the methods, I think we need to just stick with the maybe low resolution one, because that actually there. So what I mean by why I'm saying is low resolution map is I'm thinking about the bias variance trade-off. For getting, I mean, the bias variance trade-off is a really kind of famous phenomena in machine learning, that where if you want to get better generalization, you might need a more bias. That means even so you sacrifice some accuracy in your data set, but you are gonna get the better generalization, because bias model will kind of be insensitive to variance across those data sets. So actually the one really fun fact is the MPS is based on the data with 1.5 Tesla. We didn't even use a 3 Tesla data, because and it works. I mean, people have been asking how that really maybe the low resolution and then not that good signal, maybe the data is that doesn't have like really good signal, but how that works. And then I think one just the explanation is bias variance trade-off, that maybe an MPS model is really biased, but just utilize the really coarse signal that is just the gist of the pain signal, and then maybe that kind of generalized to many other data sets and many acquisition parameters. But I mean, I don't think the MPS can be used in clinic, because the accuracy there is a compromise, and I think if we really want to push the model to be used in clinic, I think each clinic maybe using the method that like, for example, phantom based optimization type of thing, and also any methods that kind of quantification of the quantitative kind of get the quantified kind of measure of fMRI. So I mean, just some absolute kind of reference. We need to establish the reference and then optimize. So that way, I think that we can give each clinic or each location or each acquisition parameter, we can give them the best accuracy, maybe biased towards that center. So I think that is the future I kind of envisioning.
I see. Okay, so so one like other question that I would like to ask is basically we are basically our group is not only us but many people here work with animal model, like animal pain model, and there's a lot of controversy. I mean, like you want to establish some pain marker. I mean, maybe will there, like, let's say, can you maybe will be a chance to find like appropriate MPS for like animal pain model? I mean that, and so really, do you really need to awake animal for that, or like anesthesia? I mean, there's other complication in animal pain model too, right? So I was wondering. I mean, because I really enjoyed your last talk in the SPA meeting before. So when you mentioned about working on like preclinical, like fMRI on animal model, like in collaboration with Professor Tokpeo, right?
Yeah.
So I was wondering, you know, are you envisioning to kind of try to come up with this MPS for preclinical animal model too, or is it? I mean, I kind of can you share some of your kind of idea how you're gonna try to approach it in treating like animal model, you know, marker for using fMRI, obviously.
I think that's a super important, I mean, key question actually. So I mean, there are many issues, and so I mean, I know that the really people really want to develop marker of pain in animal model because they only rely on the behavioral observation or just like physiological measures to define pain. So but anesthesia is a big problem, and also even in the awake animal, we are not sure they are in really pain, because pain, by definition, pain is really subjective feeling, and then if we cannot really get their report, how can we know they are in pain? So it's kind of conundrum, but at least I think we can. I mean, I always think that we have to have a kind of pragmatic kind of approach that we could at least we can get the the brain map, for example, fMRI, that is respond to the stimulus intensity, because we know that stimulus intensity, the variance of stimulus intensity, so that we can at least develop a kind of decode the stimulus intensity based on the brain, even though we don't actually have the we cannot really measure the pain. The pain is, let's say, the ground truth. So yeah, that is what I'm actually doing. So with collaboration with Professor Sungji Kim's lab, and then what we did is we actually measure the rhesus monkey's brain in the anesthesia, but giving some electric shock, and then varying the stimulus intensity, and then we actually looked at the which brain regions and really follow track the intensity, and basically we use the same predictive mapping approach. So in that way, I think we can do, but I think still kind of limited. And so what I think is in animal, and even in human, I don't think so. I mean, there are so many issues, like can we really say this is a pain signature or pain? I mean, even though we call it pain signature, there has been a lot of debate, because I think non-neuronal contribution is there even in the MPS. I think there should be some non-neuronal contributions, but no matter what, I think if we can decode the pain accurately, I think we can use this signal. And in the future, maybe we have to disentangle all these signals, and we need to know the neuronal signal. We need to isolate the neuronal signal, but that's only for, I think, for understanding the mechanisms, not like not for clinical use. I think for clinical use, for practical kind of purpose, we can use any signal we can use. So so it's kind of really hard to say what is the pain really signature in this model. But so what I'm saying is, first we need to take the pragmatism in this approach, like in biomarker development. We have to keep this dual mode. Like one is really about the mechanism mode. So we have to address reveal. We have to put our effort to reveal the mechanisms, studies mechanisms, biology, and then understand this like mechanistic kind of pain processing. And then on the other hand, I think we need to focus on application. That is not like that's quite different mode, I think. So I'm doing both. And then for animal, I think we have to focus more on the mechanistic kind of part, mechanism mode, that we need to focus more on the kind of studying the mechanisms. So I actually kind of not sure what is the real benefit of kind of developing pain marker in animal model actually. So in animal model, maybe if I kind of have the opportunity, maybe might want to do more like the circuit research instead of the marker development, because marker development, I think the goal is really pragmatic. So
I see.
Okay. I'm not sure my answer is good, but I'm still like I don't have like the definitive attitude or definitive opinion about those, but yeah, just my 2 cents.
Yeah, thanks. Well, so thank you again for your enlightening talk to Promised Land, I guess.
Thank you.
Thank you.
Good question.
I mean, my question, probably I can ask my question later, but probably other people can ask too. Yeah.
Yeah, I think that was a good discussion. Maybe others. Professor Jae-Yun Hwang from Digis, do you have any comments or question?
Yeah, so actually, the Professor Wu talked about the crisis in the reproducibility and repeatability. Why do you think that kind of issue occurs?
Why that crisis reproducibility, you mean?
Yeah.
Why that happens in the field, you mean?
Yeah. So actually, is there any the region such as the human is a very heterogeneous characteristic, has a heterogeneous characteristic. Also,
Yep.
the animal also has that kind of characteristic because of that kind of reproducibility and repeatability does not be guaranteed probably. So that kind of region can explain that kind of issue.
Oh yeah. I mean, the reproducibility crisis is not only in human studies or not only, so like even in AI area, the reproducibility crisis has been discussed there. So I think the fundamental issue is like the pressure of publishing good results. So there is a So people only report the, I mean, they can report the significant results, and then the paper is the measure of success. Actually, that is basically the currency in academia. So people really want to publish papers, and then they are eager to produce really significant results, and the p hacking is really famous as a reason for the reproducibility crisis. So people are like hacking get the p-value less than 0.05. So the p-value distribution across the whole studies is really around the 0.05. Anyway, so that is a like fundamental issue, but and as I said, some low effect sizes, like low signal to noise ratio, like, so that is really especially problematic in human studies, because human behavior is really variable, and as you said, heterogeneous. So because of that, the signal to noise ratio is really low. So because noise is large, but at the same time, I think we have to think about maybe if we know more about the factors, that if we can figure out more contributors to this noise. I mean, noise, basically variance is the signal we are dealing with, right? We need variance, but if we don't have any factors that accounting for those variance, we lose those variance as a error term. So I think that's another big issue, that we need a better theory. And so in the future, I think we need a better approach and also better theory. And I think the predictive mapping approach is one way of solving that, because as I said, the predictive map, the marker, that's itself is a hypothesis, or itself is a model or theory. I mean, theory, abstracted version of theory. So just what I mean by theory, what I mean is there is no wiggle room of this changing, no flexibility in defining this map. So I think that way we can really figure out the actual kind of pin down the actual variance we can explain with these models, and then getting better theories, and so just getting a way forward. So that's also my 2 cents. It's not complete answer, but
yeah, thank you. So
Thank you so much.
So I guess probably it's time close to finish. So maybe I can ask you one question. So this talk is very striking to me in terms of you clarify some of the confusing aspects of this pain. People say pain is difficult, and there are a lot of different aspects which is overlapping. And you kind of clarify those, like social rejection pain and other types of pain. So especially related to our like research centers kind of a goal for trying to develop ways to develop a treatment strategy for chronic pain. So I want to ask you a question regarding to the types of the pain in terms of pain biomarker. And also we hear a lot about human pain study. It's very, very difficult because the way people feel and perceive the pain is, even with the same amount of stimulus, people respond differently. And potentially that leaves us some question of maybe some ion channel distributions are like different from person to person. So I want to ask you like 2 aspects of pain biomarker. First is, can we distinguish different types of pain, which you already showed some, but especially like acute pain versus chronic pain. Even within the chronic pain, I guess there are neuropathic pain and other kinds of chronic pain. Do you see any potential for distinguishing those? And the second question is, humans varying a lot and somehow, can you consider about maybe personalized ways of pain measurement from your approach? So I hope maybe to hear some of your insights.
Yeah. I mean, they are so great questions. And the first one is, oh yes, I think we can discriminate these different types of pain. I think the issue is we don't have that enough data yet, but if we have the enough data, I think we should be able to discriminate different types of pain, and then we should do that in the future, I think. So, I mean, neuropathic pain is quite different from like fibromyalgia, and then, I mean, they have really different etiology and also different phenomenology. So, I think but even in the, for example, even in the neuropathic pain patients, there will be some aspects, some shared component with, for example, chronic back pain or fibromyalgia. So like I think the way we need to go towards is the direction we have to take is we have to have some hypothesized components that explain all different types of pain. So for example, like I mean, there should be based on some neuroscience inspired components. For example, like the evaluation, like emotional components, or so like affective components and nociceptive components, and like memory components, and so many different. There should be many different components, and I think each type of pain will will have different profiles across these different components. I think that way, I think we can I mean, without so what I'm saying is we should be able to discriminate pain types, but without these models, without these theories, I think we cannot really interpret those. And then if there is an overlap, how we really know what the overlap is. So anyway, so we need a good way of interpreting these all kind of differences and overlaps. And then, but I think we can, with a really large data set, I think even without the theory, we can do something, some kind of neuroimaging based kind of classification of different types of pain. And also the personalized one, I think that really important issue. And actually one paper is now under review on that topic. So what we did in the paper is we developed one model per person, and then we looked at where in the brain has a really large individual differences. So whether, I mean, which region is influenced by context a lot, or versus which region is really show similar patterns across people. So that shows really the results as expected. So for example, the pain, like spinothalamic kind of pain track, the related regions like the somatomotor areas and also thalamus and so on, they show really high similar patterns across people. But frontal lobe areas like the VMPFC or lateral prefrontal cortex, they show really different patterns across people. And also even within one person, they show different patterns across time. So I think, it really, we need to figure out which brain, how we can understand these individual differences, and also even within a person, what is the kind of variability, the kind kind of the source of variability across across time, for example. And so the other approach we are taking is actually we are scanning one person again and again, like that we call extensive sampling. So and then so I kind of the question we wanted to address is how accurate we can get, how well we can classify if we get a lot of data from one person. Maybe we can apply the deep learning model for that, because so far, even though we wanted to apply these deep learning models on our data, because of the data size, small data sample, we really couldn't apply those. Anyway, so by collecting a lot of data from one person, we can maybe have the sweet spot of applying the deep learning models. So anyway, so that's another way of approach to that problem, like developing personalized pain marker for just one or two people. And maybe we can have a better idea of like, yeah, better. Anyway.
Yeah, thank you. I think that's a very very interesting, and still I guess you have already like going on to this topic, which is very encouraging. So I think it's about time, like 5:30. Let's see if there's no further question. And yeah, I think this is a great time to get your insights into this, and I hope this this collaboration or our exchange will continue afterwards. And thank you very much for your real interesting and clear explanation for today. So let's thank Dr. Wu again.
Thank you so much.
Yeah,
감사합니다. 예.
네, 감사합니다.
감사합니다. 예.
감사합니다.
직접 뵙겠습니다. 예. 감사합니다. 예.
We'll look forward
감사합니다.
to interact
Yeah.
with you further in the future. Thank you so much.
You too.